Myoglobin and hemoglobin are oxygen-carrying proteins in our body. They have very different amino acid sequences but similar 3D structures. Myoglobin is a monomer with only tertiary protein structure. It has a single binding site for oxygen, which follows the Michaelis-Menten kinetics. Myoglobin functions in oxygen storage in muscles. Hemoglobin is a tetramer with 2 alpha and 2 beta subunits, each with an oxygen binding site that are subject to allosteric regulation. Hemoglobin functions in oxygen transport in blood. This video will compare the oxygen binding behavior between myoglobin and hemoglobin to explain their specific roles. Oxygen is poorly soluble in blood and therefore requires specific oxygen-carrying proteins for transport. Both myoglobin and hemoglobin binds oxygen through a prothetic group known as heme, which consists of a complex organic ring structure, protoporphyrin, bound to a single iron atom in its ferrous state. The iron atom has 6 coordination bonds, 4 to nitrogen atoms that are part of the flat porphyrin ring, and 2 perpendicular to the ring. One perpendicular coordination bond is occupied by a side chain nitrogen of a histidine residue, sequestering the iron atom slightly below the plane. When oxygen binds on the other side, the paramagnetic iron becomes diamagnetic, resulting in a color change from dark purple of deoxygenated blood to bright red of oxygenated blood. The interaction between hemoglobin or myoglobin and oxygen can be described quantitatively as protein-ligand interactions. The binding of protein, abbreviated as P, to ligand, abbreviated as L, results in protein-ligand complex, abbreviated as PL. The association constant Ka is defined as the concentration of PL over the concentration of P times the concentration of L. It is more common to consider the dissociation constant Kd, which is the reciprocal of Ka. The fractional saturation theta is defined as the binding sites occupied over the total binding sites, which equals to the concentration of protein-ligand complex over the total protein concentration, which is PL plus free proteins. It is equivalent to the ligand concentration over the sum of ligand concentration plus the dissociation constant. From this reaction, we see that Kd equals the ligand concentration at which half of the available ligand binding sites are occupied. In the case of hemoglobin and myoglobin, Kd can be replaced by P50, the partial pressure of oxygen when half of the available ligand binding sites are occupied. Therefore, fractional saturation is equivalent to partial pressure of oxygen over the sum of partial pressure of oxygen and P50. The binding curves of myoglobin and hemoglobin plots fractional saturation against the partial pressure of oxygen. Myoglobin exhibits a hyperbolic binding curve, shown in orange, whereas hemoglobin exhibits a sigmoidal binding curve, shown in green. The dissociation constant for myoglobin is much lower than hemoglobin, which means that myoglobin has a higher affinity for oxygen than hemoglobin. This difference is due to the fact that hemoglobin is an allosteric protein while myoglobin isn't. For an allosteric protein, the binding of a ligand to one site affects the binding properties of other sites of the same protein. Each of the 4 subunits of hemoglobin exhibits 2 distinct conformations, the T state and the R state. When oxygen is absent, the T state, which stands for tense state, is stabilized by salt bridges and has low affinity to oxygen. The binding of oxygen disrupts the salt bridges and triggers a change in conformation to the R state, which stands for relaxed state and has high affinity to oxygen. At high oxygen concentration in lungs, hemoglobin is in high affinity R state, which binds tightly to oxygen. At low oxygen concentration in the peripheral tissues, hemoglobin transitions to a low affinity T state, releasing the bound oxygen, thus facilitating its role of oxygen transport between the lung and peripheral tissues. On the other hand, myoglobin still exhibits the high affinity R state in peripheral tissues and would only release oxygen when the concentration drops really low, thus facilitating its role as oxygen-storing molecule in the muscles. The transition of T state to R state in hemoglobin can be explained by cooperativity, in which the binding of oxygen to hemoglobin increases the affinity of remaining unfilled binding sites. Hemoglobin is subject to various allosteric regulation. A homotropic modulator acts as a ligand, whereas a heterotropic modulator doesn't. Oxygen acts as both a substrate and a homotropic activator. As mentioned earlier, the binding of oxygen stabilizes the R state, increasing the affinity of other oxygen binding sites through cooperativity. Carbon monoxide acts as a homotropic inhibitor that competitively binds to ferrous iron stronger than oxygen. Therefore, carbon monoxide is considered toxic. Both proton and carbon dioxide act as heterotropic inhibitors of hemoglobin that stabilizes the low affinity T state. Carbon dioxide is produced by catabolism of organic fuels in mitochondria. It is hydrated by carbonic anhydrase, abbreviated as CAH, to form carbonic acid, which spontaneously dissociates into proton and bicarbonate. Protons bind to a specific histidine residue of hemoglobin, stabilizing the low-affinity T state, shifting the binding curve to the right. This phenomenon is known as the Bohr effect, which helps hemoglobin release even more oxygen to peripheral tissues when metabolic activities increase carbon dioxide concentration. 2,3-bisphosphoglycerate is another heterotropic inhibitor of hemoglobin that stabilizes T state and shifts the binding curve to the right. Adult hemoglobin, abbreviated as HbA, contains a higher concentration of 2,3-bisphosphoglycerate than fetal hemoglobin, abbreviated as HbF. As a result, a mother's hemoglobin would have lower oxygen affinity than the baby's hemoglobin, leading to successful transfer of oxygen from the mother to the baby. On the other hand, 2,3-bisphosphoglycerate is also important in the physiological adaptation to lower oxygen concentration at higher altitudes, known as hypoxic conditions. Low oxygen rapidly increases the concentration of 2,3-bisphosphoglycerate, resulting in a decreased hemoglobin affinity for oxygen, and therefore more oxygen is released from hemoglobin and available for peripheral tissues.